In our second lecture on the survey of herbicide mechanism of action, we will talk about inhibitors of biopolymer synthesis. Now, what do I mean by this? Um, Other than photosynthesis, plants make a lot of molecules, either making building blocks or they assemble into larger molecules. And so, for example, sugars are synthesized and can be put together into cellulose. Some fatty acids can be made, elongated, and form into membranes and cuticles. And amino acids can be made by different pathways, and their assembly into proteins will form larger molecules. So the first one we'll talk about will be inhibitors of cellulose biosynthesis, which is group 29. And that's a fairly new understanding of how they work. So first of all, the synthesis of cellulose happens on the surface of the plasma membrane. So that's the plasma membrane. And you have these six-membered protein that makes a rosette. That's a cellulose synthase complex, CESA. You'll see that on the slide as CESA. That's cellulose synthase complex. And this is actually a SEM of the surface of the plasma membrane, where you can see actually the proteins lined up just like this, like as a rosette. And there's three groups of those, and they all inhibit cellulose biosynthesis, but via different mechanisms, and people don't really understand. But the group one removes the CSA, the cellulose synthase complex. The group 2 accumulates cellulose synthase, but they slow or stop the movement. So these proteins move across membranes, and these inhibitors actually stop the movement of the cellulose synthase complex. And the group 3 changes the direction. So instead of being lined up very nicely, the cellulose is disordered. And typically roots tend to swell, tissue expand because the cells cannot divide properly. And then there's reduced cellulose biosynthesis and incorporation. The other group will be in the fatty acid groups right here. So ACCAs, acetyl-CoA carboxylase, is the first step in fatty acid biosynthesis. And these herbicides tend to be applied post-emergent. There's a lot of foliar absorption, and typically esters are made. So those herbicides have ester groups that makes them more lipophilic, and they get into the cuticle, and then they are converted back to the acids to inhibit the targets. They have some flow and mobility, and they translocate to the meristematic tissue. I mentioned that about the esters that affect their ability to take up in the plant, and they tend to translocate to the meristem in the roots, because this is where a lot of lipid biosynthesis happens. And when that's inhibited, the plants die. They die very slowly, and it takes 7 to 14 days for the symptoms to develop, even though root and shoot growth stops. So the plants stop growing, but they don't die for 7 days or up to 14 days. One of the key symptoms is that you can remove the stem from the leaf in grasses. So these are grass-specific herbicides. And why are they grass-specific, meaning that they only kill grasses? Is that in plants, there are two forms of that enzyme, ACCAs. There's a prokaryotic form, which is multi-subunit made out of 3 different proteins. And there's a eukaryotic form, which is multifunctional. It's one large protein with all 3 functions. So here there's 3 enzymes, each with their function separately put together, and here's one enzyme with the 3 functions put together. What's interesting is that in dicot cells, the plants that survive the treatment with these herbicides, they have the eukaryotic in the cytosol and the prokaryotic in the chloroplast. And the prokaryotic form is not sensitive to the herbicide. And this is where most of the fatty acid biosynthesis happens. In grasses, they only have one form, both in the cytosol and the chloroplast, and that's the eukaryotic form, the one large protein. And that form is actually sensitive to the herbicide. So when you put these ACCase inhibitors, you kill grasses because you kill all of the enzymes, and the dicots survive because these in the plastids are not affected by the herbicide. Another target of herbicide that affect lipid is the elongases. And so once fatty acids are made, they can be made longer by enzymes called very long chain fatty acid elongases. And there are several enzymes. And what happens is these herbicides inhibit several of these target sites. So one herbicide will affect several of these elongases. Symptoms are very similar, very slow death of the plant. However, these soil applied they are soil applied rather than foliar applied like the ACCase inhibitors. They require high rate. They are slow acting, and because herbicides target different enzymes, it's not likely to evolve target site resistance through site mutations because you would need mutation several genes before the herbicide will stop being effective. Now moving on to the main class of biopolymers, which will be the fatty acids. The proteins. And so there's three key targets for these: EPSPS in the aromatic amino acid biosynthesis, ALS in the branch chain, and then GS in glutamine. And we'll talk about GS in the third lecture because it's kind of a special case. So the EPSPS inhibitor, that's glyphosate, is group G, and now it's actually called group 9 according to the new classification. And it's the only herbicide in this class. That's glyphosate, and it's a non-selective herbicide. It's the most abundantly used herbicide in the world. It's rapid foliar absorption, translocation to all parts of the plant. It's used in many non-selective situations as well as in glyphosate-resistant crops because it's non-selective. It will kill pretty much all the plants. The rate is pretty high, 400 to 600 grams in GR crops, 800 to 900 grams. It's most used post-emergence. It's water soluble. It's applied on the leaf. If it hits the soil, there's no soil activity at all. And the activity is pretty slow. It takes a week to 10 days for the plants to die. The biosynthesis, the enzyme being targeted is this enzyme right here that's involved in the synthesis of phenylalanine, tyrosine, and tryptophan, these aromatic amino acids. And the enzyme is nuclear encoded and is located in the chloroplast. And this reaction is pretty complicated. One of the substrate, this one, binds first. It creates a binding site for PEP. PEP binds next. That leads to the reaction in EPSP. The product of the reaction is released. That is then used to make these aromatic amino acids. Well, glyphosate inhibits EPSPS, that enzyme that catalyzes this reaction right here. And that blocks the biosynthesis of aromatic amino acids, and that leads to slow death of plants. And then the last herbicide class we'll talk about in this lecture will be the ALS, involved in branch chain amino acid. That's group 2, or formerly group B. This is a very important class of herbicide as well because they were the first herbicides used at very, very low rate. So when they came out, it was a big revolution in herbicide because instead of using kilograms of herbicides, people use grams of herbicides. And it inhibits ALS. A couple of herbicides mentioned here, but ALS, they are foliar applied, but they can also be root absorbed. They have broad spectrum activity, which they control a lot of different species, not as broad as glyphosate, but quite a bit. Translocate to the shoot and root meristem. And same as fatty acid biosynthesis, the plants stop growing. There's not much symptoms happening at first, but after 7 to 10 days, the plant will die. However, this class of herbicide is extremely sensitive to herbicide resistance. As I mentioned, so this enzyme is involved in branch chain amino acid biosynthesis. It starts with pyruvate, goes to valine and leucine on one branch, and the other branch starts with 2-ketobutyrate and end up with isoleucine. And these pathways go in parallel, and one enzyme, ALS, is right there and catalyzes both of these reactions, leading to these 3 amino acids. Many herbicides have been developed to target ALS. And that blocks, of course, the synthesis of branch chain amino acids. Slow acting, some malformation and stunting are symptoms. Resistance, I mentioned, is very common and it can be very strong. Here, the 1x completely kills the plant. And here, we're talking about 100x, and the plant is maybe only 20% inhibition. So the level of resistance is tremendous. And the reason why that is because if you look at the enzymes, there's a dimer formation, and there's a channel opening here that leads to the inside where the activity of the enzyme happens. But the herbicides don't bind in the catalytic domain. What they do is they bind right there at the interface of that, in the pocket right here between those two. So that blocks access to the catalytic domain. And there's a bunch of different mutations that can be done by plants on these area that provides resistance to the herbicide without affecting the activity of the enzyme at all. So this is the end of our second lecture on herbicide mode of action.